Now that we've considered the structure and properties of amino acids, let's consider the primary structure of their polymeric form. Proteins that are biologically active are composed of a series of amino acids linked by covalent peptide bonds. For any given protein, there may be many three-dimensional conformations possible due to the flexibility of bonds within the protein. However, the native conformation will be that which confers biological activity. In the figure here, we see a representation of the native conformation of myoglobin, which is vital to its function as an oxygen-binding protein. Portions of the native conformation may be comprised of large segments that contain no repeating or recognizable structures. However, these segments are not truly random in the sense that the native conformation always includes these segments. In other words, the protein always folds in the same way. There are three levels of protein structure that are present in all proteins, and a fourth level in some. The primary structure is simply the order of the amino acids as they link together in the chain, reading from the N or nitrogen to the C or carboxy terminal. The three-dimensional arrangement of the backbone atoms makes up the secondary structure of the protein. Tertiary structure is the final fold of the protein in three-dimensional space, including the position of atoms in the backbone, side chains, and any prosthetic groups. Quaternary structure is only present in proteins that are comprised of more than one subunit or polypeptide chain. The quaternary structure is the arrangement of these subunits with respect to one another. Let's focus now on the primary structure of a protein. Recall from an earlier lesson that the order of monomers in our polymer is informational. That is, the order determines the final structure and function of the molecule. We'll look at how to determine the amino acid sequence of a protein in a separate lesson. A good example of the importance of amino acid sequence is in the case of a mutated form of the oxygen-binding protein hemoglobin, which is responsible for delivering oxygen from the lungs to the tissues. A mutated form of this protein, known as HbS, is the causative factor in producing sickle cell anemia. The mutated protein carries a single amino acid change, which replaces the wild-type glutamate residue with that of valine. We'll look at this in greater detail later, but you can see the effect of this mutation from the figures given here. Notice the sickling effect due to the mutant form of this protein. This single change has a powerful effect not only on hemoglobin structure and function, but also on the structure of the erythrocytes or red blood cells that carry the mutant protein.